Okay, welcome. The key research by Raine. So this fits into the biological area, and it is a biological or physiological explanation of criminality. So, brain abnormalities and murders indicated by positron emission tomography or PET scans. So we are essentially, before we start, we're essentially looking at a study where the PET scans show a difference between murderers versus non-murderers in the particular areas of their brain that show activation or not. There is Professor Raine, quite a controversial guy in some ways, and that's why this research relates to the issue of social control. It opens up an awful lot of ethical questions of whether we withdraw people from society or not based on their brain function. I always remember him as Raine, sounds like brain, and he is examining the brain. So there we go. Right, theory or theories on which the study is based. This is where the eyes are in the human brain looking forwards, for this brain it's looking the opposite direction. So these are two people looking at each other. There's the brainstem at the top of the neck. The hippocampus, which we're aware of from the Maguire study, in the middle there. We've got the corpus callosum that's in the middle, joining the two hemispheres. We know that from the Sperry study. Got the amygdala, just back here, the anterior cingulate cortex, and the prefrontal cortex. Now, a little tip for you. Actually identifying these areas of the brain isn't necessary for the study. You really just need to learn a few examples of some of the areas where there was a difference between the murderers and the non-murderers. So don't worry too much about being able to locate them in your own brain. So, brain dysfunction. The idea, in terms of a theory, that damage to the prefrontal cortex might result in impulsive behaviour, immaturity, altered emotionality, loss of self-control. So altered emotionality might be being more aggressive. It might be getting upset quicker than other people would. Loss of self-control is, you've got these impulses, but you can't stop them. Inability to modify behaviour. If you link to Casey study, where we should have higher levels of control in our frontal gyrus, then those are the kind of things we're talking about here. All of these can increase the likelihood of aggressive acts. So the amygdala is associated with aggressive behaviour. So along with the recognition of emotional stimuli, like being able to tell when someone is scared on a person's face. So damage to the amygdala could be associated, or has been associated, with fearlessness and abnormal functioning. So learning emotional responses, appropriate emotional responses, might not be possible if we have damage here or faulty activity here. So whereas we might see someone's face looking scared, that's a scared face, we might say, oh, I'm not going to push them any further. You would empathise with them. Whereas if you're not picking up on these signals, then perhaps you're not responding in what we'd regard as a normal way. So we might continue to scare this person or not make them feel better, or we might not register the fear in their face. One particular group of offenders are those who plead not guilty, NGRI, not guilty for reasons of insanity. So you've all heard of these. Sometimes people say it's people trying to get off, which I'm sure it is in some cases. In other cases, are they insane? And Raine's study is based on results found through the PET scans. They were referred to Raine by lawyers or the courts to try and see if they were classed as insane. So could they plead guilty for reasons of insanity? So PET scans involves patients being injected with a harmless radioactive sugar substance, which is known as a tracer. The most active brain tissue uses the glucose and attracts the radioactive substance. Radiation sensors in the scanner detect where the radiation is greatest, and builds up a picture of activity in the brain. The scans takes between 10 and 40 minutes to complete and are painless. The data from the scan is usually presented on a coloured picture, where hot colours, like orange and red, are used to represent the greater activity of the brain, whereas the cool colours, blue and green, represent areas of least activity. If I just quickly skip forward a second to show you a picture of that, there we are. So, red, most activity has been going on there. The tracer attaches to the glucose. Attached to the glucose in the blood. So the thing that you're injected with attaches on, and then when that's used up in the brain, all that's left is the tracer. So if someone's been doing a lot of thinking, then you would see that lots of that sugar, lots of that glucose has been burned up, and so this person has probably been thinking for the last 20 minutes or so. Okay, so they're comparing normal and murderers' brains to see the difference. So you can see there, just to get ahead of ourselves, but you can see some big differences in that particular slice of the brain. So let's bob back. So Raine uses PET scans to discover the brain abnormalities, and we've mentioned all this. Until this study, no previous brain imaging had been conducted to support or challenge the notion that people could plead not guilty for reasons of insanity. So people who plead that are more prone to violence than non-murderers. That's the argument. So Raine is trying to draw this correlation really between these areas of the brain that have previously been associated with aggression. For example, in research where someone's had a tumour in a particular area of the brain and they've suddenly become much more aggressive, or the opposite, completely unaggressive, then he's linking this together to say, right, well, maybe this could be a brain functioning reason behind why the NGRIs, they're not guilty for reasons of insanity, might have been more aggressive than the norm. So I did a pilot study involving 22 NGRIs compared to 22 normals, and Raine in 1994 showed support for the idea of prefrontal dysfunction in the NGRIs. So prefrontal is the front of the brain, dysfunction is abnormal functioning of the brain. So the aim was to build on the previous research, and two hypotheses were tested. Seriously violent individuals pleading NGRI will have relatively localized brain dysfunction, so particular areas of the brain is what local means. In the prefrontal cortex, angular gyrus, amygdala, hippocampus, thalamus, and corpus callosum. So again, you don't need to know particularly where they are, but remember a few of those for your exam. Seriously violent individuals pleading NGRI show no dysfunction in other brain areas which have been implicated in other psychiatric conditions, but which have not been related to violence. Okay, so they're not going to show areas, they're not going to show any differences in those other areas that that haven't been related to violence. So you're looking for a specific localized areas just for the violent areas. Research method, quasi-experiment. The IV is naturally occurring. I should just say, nipping back there, I didn't quite explain that well enough. Right, the psychiatric conditions, they were matched when we get to this. We have a matched pairs design with things like schizophrenia. So a schizophrenic murderer was matched with a schizophrenic non-murderer. Now, so in their brains, you are going to see areas of the brain associated with schizophrenia that are different to control groups. But what Raine's saying is you won't see any difference between the schizophrenic murderer and non-murderer in these areas that aren't associated with violence. So, for example, areas of the brain for auditory hallucinations, you're going to see abnormal functioning, but equally abnormal functioning between these two compared to people who don't have schizophrenia. But because that isn't associated with violence, then you're not going to see a difference between it. if you see what I mean. That's the point of doing a matched pairs design, is you can say, well, that different area of the brain is because they've got schizophrenia. It's not because they are violent. Okay. So, quasi-IV naturally occurring of NGRIs or not. MPD, the matched pairs design, matched on age, gender, and 6 schizophrenic non-murderers with 6 schizophrenic murderers. IV is whether the participant was pleading not guilty for reasons of insanity or a normal non-murderer. DV, whether the participant showed evidence of brain dysfunction in the prefrontal cortex and other areas such as those that we named earlier that have been previously linked to violent behaviour. Controls, no medication, so they were asked to stay medication free, no history of psychiatric illness other than the schizophrenics, or current medical illness. So, sample experimental group of murderers consisted of 41 murderers, 39 males, 2 females. Mean age of 34.3 years. Tried in the state of California and charged with either murder or manslaughter. Sent to UCI, Imaging Centre, for one of 3 reasons. So again, the lawyers have sent them, or the court has sent them, to obtain evidence as to whether they are NGRI or not. Obviously, it's going to help your defence if you can say a scientist has said that you have abnormal brain functioning. To find out if they were competent to understand the judicial process, so is this person's brain so abnormal that they can't even understand what they're being tried for, that would have an implication of whether you went to prison or psychiatric hospital. And to see if there are any evidence of diminished mental capacity which may have affected the nature of the sentence that they get. So again, a prison, it could be the length of sentence, but it is again more likely to contribute to, even if they can understand the judicial process, was there some reason why we should be sending them to a hospital rather than a prison? They were referred for the following reasons. 6 had schizophrenia, 23 had head injuries, 3 had a history of drug abuse, 2 had affective disorders, so that's like depression, 2 had epilepsy, 3 had a history of hyperactivity and learning difficulties, and 2 had personality disorders. So, therefore, when they turn up, the lawyer is going to say, for the defence, is going to say, hang on, you've got one of these histories, let's send you to the imaging centre and see if there's any brain functioning reason why we can add to your defence, how we can add to your defence. Perhaps we can get a lesser sentence, perhaps we can get to a hospital, but perhaps we can just understand more of why you did what you did. In 7 of the above cases, there was also unusual circumstances surrounding the crime that had led to suspicion of mental impairment. I'll let your imagination run wild with that, but it would be something abnormal to a normal murder. Perhaps something that indicated that this person had mental difficulties. Control, non-murderers consisted of 41 participants, so matched age, gender, schizophrenia. The rest were thoroughly screened to no history of psychiatric illness. Procedure, the materials, a thermoplastic head holder, so something that held their head in place, moulded to the participant's head while they were being scanned, a PET scan, a fluorodeoxyglucose, FDG I'll refer to it as in the future, tracer injected to trace brain metabolism, so that's clinging onto those glucose. Continuous performance task, so that gets them to detect target signals for 32 minutes. All that is, a continuous performance task, is a screen in the scanner. They've got to watch it and they might have to click when they see particular images, but it means that you are getting them to do the same task as the other participants at the same time. All offenders were in custody and kept medication free for 2 weeks. Scanning 10 minutes before they received the FDG injection. All participants were given a practice trial on the CPT, on the continuous performance task. 30 seconds before the injection, they started actual continuous performance task, so that the initial novelty would not be FDG labelled. In other words, that it wouldn't be, the novelty of it wouldn't soak up all the glucose. You're getting their brains working and used to the task just before the injection. 32 minutes after the injection, the participant was transferred to the adjacent PET scanner room. An individually moulded thermoplastic head holder was used to keep their head in place, and 10 slides at 10mm intervals were taken both outside and inside the brain, so taking pictures through the brain. We'll cover the results in the next video.